All right, guys. So go ahead and title your notes carbon. We're going to start with a discussion on carbon. And the first point that I want to make is that life is carbon based. And an organic molecule is a molecule that comes from a living thing. So when we look at all these molecules, that you can assume that they're all produced by living things. And before life, you would have never found any of these organic molecules on the earth. Like we can imagine a time when the earth had nothing living. It was just kind of like, right, uninhabited. And at that time, you would have never found sugars, you would have never found lipids, carbohydrates, proteins. Those molecules just didn't exist because you have to have a living thing to make it. All right. Comes from a living thing, and these molecules are carbon based. Now, why carbon? Why is it carbon based and not, let's say, boron based or sulfur based? Or, right? I mean, it could have, of all those elements, there's a lot of elements that we could have based life on, but life is based on carbon. All right, so the reason for that, carbon is very versatile. So carbon has a valence shell with 4 unpaired electrons. And we're going to draw that real quick. So that would look like this, where the inner shell has 2, and then the outer shell has 4 lone electrons. So what this means is that carbon is like totally ready to connect to other atoms. In fact, carbon wants to form 4 bonds, 4 new bonds, in order to find stability. So nature prefers stability over instability. So this is a general rule. Things will adapt in order to find a stable state. And carbon, this is true for carbon, in order to find stability. This gives it great versatility. You can get a lot of different shapes and bonds and with carbon. Carbon can form chains. It can form rings. It can form single bonds, double bonds, triple bonds. It just has a lot of versatility, and you'll see some of that in a minute here. So, for example, you could have a carbon atom right here that joins up with 4 hydrogens. So you got a hydrogen here, This is going to join up. This hydrogen is going to join up here. This hydrogen is going to join in here. And then this hydrogen will come in and join here. And that's going to produce this methane molecule. And now the point that I want to make here is that a methane molecule, CH4, is a much more stable situation than, say, just a lone carbon atom. So that's just one example. It's really, really simple organic molecule. And okay, so methane, this is a example of a hydrocarbon. Okay. Now, if we break down the word hydrocarbon, we find 2 parts. There's the hydro, and then there's the carbon. Hydro means hydrogen, carbon means carbon. So this is any molecule that's made of carbons and hydrogens. And there are just countless examples. There are so many examples of molecules that are made of just carbon and hydrogen. It's like really easy to form those kinds of molecules. So a hydrocarbon consists of only hydrogen and carbon. Okay. They can form hydrocarbons can be chains, or which another word for that is aliphatic, or the molecule can be aromatic, and that would mean that it takes the form of a ring. Wow, I can't spell. Aromatic. And actually, it's kind of interesting. The reason that it got the name aromatic is that usually when the hydrocarbon is in a ring structure, it smells like it's got an aroma, and I'm not entirely sure why that is, but a lot of the aromas that that we perceive are little aromatic rings, hydrocarbon rings that are quite literally floating in the air, and when you inhale it, the molecule binds to like a little receptor in your nose. So that's how they got the name aromatic, those rings. So I would draw examples, but I don't think we need to. Just see the reading for that. You'll see examples of rings versus chains. Anyway, okay. So actually, we're going to draw a ring right now. What I'm going to show you guys right now is this concept of there's two kinds of reactions that can take place between organic molecules. And but first, let's just state this, that macromolecules, we're about to study 4 different types of macromolecules. Macro means large, right? So these are big molecules. Macromolecules are polymers. And if you break down the etymology of the word polymer, poly means many, and mer means part. So the literal meaning of polymer means many parts. And usually those parts are repeating, like they kind of link up and they just repeat. Now we're going to say that polymers are formed from a reaction called a dehydration synthesis. Now you guys definitely need to know this. Definitely. It's just like a really, it just comes up a lot. Dehydration synthesis. So I'm going to underline that dehydration synthesis reaction. And if you think of what does that mean? What does dehydration mean? Well, dehydration means if you break down the etymology of the word, it means to remove water. And that's quite literally what happens in this reaction. A water molecule is removed from the organic molecule. It's like stripped off of it, and then a bond is formed. So what would that look like? Well, let's take the simplest example we can. Picture and this is our first example of a ring. Okay, we said that hydrocarbons can form chains or they can form rings. Example of a ring is this glucose molecule. Looks like that. So what if you took 2 glucose molecules and put them next to each other? Will they react? Will they react with each other? Will they connect? The answer is yes. If the conditions are right. Oh no. Get to class. They're you're laughing. You know those people? Yeah. Are they do you like them? Are they good friends to you? Good. That's good. They're funny. They were a fun group. They sat like right here and they all sat together. It was crazy. Too crazy. But anyway, let's say that these are each glucose molecules. Yes, and these are, by the way, what we would call a monomer. So right now that they exist as monomers, right? They're just one part. But if we join them together, uh, okay, this is where we got to pay attention. So we said that we're going to remove, in other words, we're going to produce a water molecule. So I want you to circle right now. I want you to circle this hydroxyl group. This is called a hydroxyl, the OH. Circle that. That whole thing is going to get removed. And then what do we need? If we're going to produce a water, what else do we need? We got one hydrogen and one oxygen that are going to go toward the water. So what do we need to take from here to make a water molecule? Do we need another, like one more oxygen? Do we need two more oxygens? Do we need one more hydrogen? Do we need two more hydrogens? What do we need? Let's do the math in our heads here. Shout it out. What do we need? You guys got this. Don't be shy. We need an oxygen because hydrogen is HO2. No. Water is H2O, which means that, you guys just saw the water molecule on the board for the warm-up question. There's an oxygen and there's two hydrogens. So far we've got one oxygen, so that's good, but we need two hydrogens. So we're gonna have to take just the hydrogen from the other one. Okay, so circle just the hydrogen here. Now, if we took those things, connected them together, that's going to produce us a water molecule, and thus the dehydration. Does everybody see that? That's why we call it dehydration. We're taking the water molecule out, producing the water molecule. So I want you to think about what's going to be the product over here. What's it going to look like? Well, all that's going to be left is two sugars and an oxygen in the center, right? So it'll look kind of like this. Is that logical to everybody? And by the way, this product over here is called maltose. So it's a new molecule. It is what you would call a disaccharide. It's a sugar that is composed of two parts, disaccharide, called maltose. And this is also considered a polymer. So notice that we went from having monomers to now having a polymer. We finally have a polymer. A polymer is something that's made of many parts. All right, now what if we, hang on. Don't forget to label this. This has a name, okay? This little bond in the middle that joins the two sugars. This bond is called a glycosidic linkage. Or actually, yeah, let's call it a glycosidic bond. I think that's the terminology they're going to use, glycosidic bond. Alrighty, any questions on that? So what if we did the opposite of this dehydration synthesis? Like what if we, instead of connecting two things together, what if we took a molecule and we broke it apart? That would be kind of the reverse, right? If you go in the reverse direction, you're going to be breaking apart. So this next reaction is basically the reverse. That would be That would be when monomers are formed from hydrolysis reaction. Now, if you look at the etymology of hydrolysis, hydro, water, lys, split. Okay, so you can kind of derive the meaning from that word just from the etymology of the word there. Now, a hydrolysis reaction would look like this. It's where you're starting with maltose, and we're going to break it apart. We're going to go ahead and absorb a water molecule from the environment. So this could be a water molecule from the surrounding solution, let's say, from the environment. And that water molecule is going to come in and join up with the sugars to produce just two regular glucose molecules. So you guys can see that these two reactions are the exact inverse of each other. This one is just the reverse of this. Like if you went this way instead of this way, right? One other thing that's kind of a little bit interesting, and I will admit I'm jumping the gun a little bit or going a little bit ahead, but this reaction where you're splitting molecules apart, it yields energy. Whereas the reaction up here, this reaction actually consumes energy. So don't worry too much about that, but we will, what is it? Unit 3. We will definitely talk about the energy when we get to Unit 3. But right now, that's all you need to know about that. Moving right along. I know today's a little bit grueling. It's like just a lot of notes, and but it's important. This stuff is foundational. And maybe you'll learn that organic chemistry is just not for me, and you're not gonna want to go into this. Maybe. I think if you're doing nursing or pre-med, anything like that, you have to take organic chemistry. It's like probably the most important class that you would take for that. It's like super important. All right, so let's look at organic molecules. We're ready to look at the categories. There's four categories, 4 main categories. And so one would be, we're going to start with carbohydrates, teach you about those. The second category that we will talk about is lipids. The third will be I think we'll do nucleic acids third. And then the fourth one we'll talk about. The fourth one is probably the most complex, and that'll be proteins. So we save that for last because it's the hardest. It's the hardest to understand. Alrighty. So let's start with carbohydrates. That will be our first molecule. All right. Now, I know I do this a lot, but I like to break apart the word. Carbon, hyd, or HYD, hydra, is water. So if you break apart the word carbohydrate, it gives you a little clue as to what it might be made of. Think of it as basically being made of carbons, hydrogens, and oxygens. in a 1 to 2 to 1 ratio. In other words, for every one carbon atom that you have, you can expect to find 2 hydrogens. And for every one carbon atom, you can expect to find, not always, but you can sometimes expect to find 1 oxygen. So often they will exist in a 1 to 2 to 1 ratio. And you're going to see some examples of that momentarily. Yeah. Okay. So let's look at some. You must know these carbohydrates. These are carbohydrates that you've got to know like the back of your hand. And that would be the monosaccharides. There are 2 that I want you to know. Now, remember, mono means single, and then saccharide means sugar. So these are sugars that are composed of 1 unit. And the first example is the glucose that we talked about. Notice that it's hexagonal structure, hexagonal shape, you could say. And then the other one that I want you to know, glucose. The other one I want you to know is ribose. Ribose is a little bit different chemically. It forms a pentagonal shape. And there's an oxygen right there. And that's called ribose. Now, what is the function of these things? Mostly energy, okay? They are a quick source of energy. But you'll also find out that ribose is structural too. There's a couple things that are made of ribose. We'll get to those later. But glucose, if you, my mom has diabetes, and so she carries glucose tablets on her all the time to help with her blood sugar, right? So the sugar that's floating around your blood is glucose, and it's like the most quickly, readily available source of energy for as far as carbohydrates go. All right, so next we look at the disaccharides. And you know that di means 2. So these are sugars that are composed of 2 parts. We talked about the maltose earlier. But let's take a look at table sugar. So table sugar is composed of a glucose and a ribose. So you've got a glucose over here on the left, and then a ribose on the right, joined together by, of course, what we saw earlier, which is the glycosidic bond. And it's called sucrose. Sucrose, aka table sugar. Now, there are several other examples of disaccharides. For example, maltose is a disaccharide. Lactose, which is the sugar that's in milk, is also a disaccharide. Function, pretty much just energy, right? It's like a type of food. Next, polysaccharides. So this is where you've got 3 or more parts. One example that we find in plants are made out of fiber. In fact, paper, like the paper that you're writing on, is composed of a polysaccharide called cellulose. It's found in the cell walls of plants. And cellulose has this structure where you've got this alternating up, down, glycosidic bond. So if you can picture these sugars that are connected together, hexagonal sugars, right? Glucose. And these glucose sugars have been connected, but their glycosidic bonds form an up, down pattern. So notice that this one is down, this one is up, the next one is going to be down. Up, down, up, down, up, down. And what that does is it creates a linear molecule. And it forms fiber, right? Specifically cellulose fiber in the cell walls of plants. That's where that is found. It has a high tensile strength, so it's very, very sturdy molecule. There's another molecule that really closely resembles cellulose, and that is starch. The difference, though, is that starch has a down, 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 down orientation of the bonding. So you've got all the bonds are just down. And the result is it actually is kind of results in a little bit of a curved shape. I didn't do a good job showing the curve, but curved shape, more easily broken down than cellulose. In fact, cellulose, I'm going to add this. Cellulose is indigestible. So have you guys ever heard of dietary fiber before? Like if you look at the ingredients on the back of, or the nutritional facts, you might see something called dietary fiber. And what that's referring to is it's referring to cellulose. And when the cellulose is in your digestive tract, it is indigestible. It'll actually pass through you and remain intact. Whereas starch will get broken down. These bonds will all get chopped up and your body will process it as individual pieces. And that's simply not the case for cellulose. The bonding is too strong. Okay, and so in terms of function of starch is energy storage. Think of a potato. A potato is full of starch, and that's actually the plant's method of storing energy. The plant stores energy in these little tubers that are underground. You guys ever dug up a potato before? You ever grown one? Kind of cool. You got this plant. This is above ground plant that's maybe 2 to 3 feet tall. And as soon as you're ready to harvest, you actually take the shovel and just start digging up. And connected to the roots are these giant potatoes. So you got the root, and then there's just these potatoes that are just attached to the roots. And that's the plant's method of storing energy. It stores it as a starch in the roots. All right. Questions about carbohydrates? Good. Okay. Now tomorrow, in the lab tomorrow, you're going to be given these mystery molecules. We're going to have all these mysterious solutions, basically. And you're going to have to do these like chemical tests to figure out if there's carbohydrates present, chemical tests to see if there's proteins present. So today I'm kind of trying to give you the groundwork here to help you with the lab that you're going to do tomorrow. So, okay, next is lipids. Take a deep breath, stretch, or whatever you got to do to keep yourself focused. Okay, so we're shifting gears now to macromolecule number 2. Remember, we just, if we go back to our list, we just got carbohydrates. We understand those pretty well now. But now it's time to look at lipids. Now, lipids, a couple things that characterize them is that they are non-polar chain. So it's going to form a chain instead of a ring. So most of the carbohydrates we looked at were these little rings, right? They don't really look like rings, but they are. And then lipids are going to be chains, kind of zigzags. And let's go ahead and define the most common lipid, which is a fatty acid. This is our first example. A fatty acid is a chain of carbon atoms with a carboxyl group. You guys probably can't read that. Let me write that a little more clear. Carboxyl group. Underline carboxyl group because that's an important functional group that I want you guys to know. All right, so what would that look like then? So the chain would look like this. So in organic chemistry, whenever you see a zigzag, that represents a chain. And every time that you see a corner of the chain will represent a carbon atom. So you can put a carbon on each corner of the chain if you'd like. Now connected to the end of this chain is going to be a carbon atom that forms a double bond to an oxygen and then a single bond to our hydroxyl group. And this whole thing right here, I'm going to draw a dotted line around it. That's a carboxyl group. And then this other piece, right, the long chain, that's the chain. So a fatty acid is nothing but a carboxyl group attached to a chain. But there is something you need to know about this, and that is that the chain can come in 2 different flavors. The chain can be saturated or unsaturated. And what that means is, I want to specify, I want to say it's saturated with carbon atoms. So what that means, what it means to saturate something is that it's completely filled up. Like let's say, I'm trying to think of another usage of the word saturate. Like, I don't know, you came home on a rainy day and your sweatshirt was saturated with water. That would mean that it was completely soaked up with water. Like it couldn't, in other words, your sweatshirt, it's not possible for your sweatshirt to absorb any more water. It is completely saturated, right? Think of these molecules as being completely saturated with carbon atoms. In other words, they can't get in, oh no, I'm sorry. Gosh, hydrogen atoms. Completely saturated with hydrogen atoms. So think of these molecules as not being able to take on any more hydrogen atoms. So what would that look like? Saturated would look like this. In other words, there are 2 hydrogens on each, bound to each carbon atom. That's what that would look like, okay? In other words, it's filled up, completely filled up with hydrogens. Now, if you are not saturated, so this is what we call saturated. saturated with hydrogens. If you're not saturated with hydrogens, you would have a double bond somewhere. So you'd be going along like this. And then you've got so right here you've got a double bond, which results in a kink in the chain. And that would mean that this is an area that's not saturated, or unsaturated is the word. Unsaturated fatty acid. And if you want, you can add a little arrow here that says we've got missing hydrogens, which means it's not fully saturated, right? Not fully saturated. And so it leaves this double bond, which kinks the chain and makes it, it's not straight. It's got a bend. There's a bend in the chain. All right, why do we care? Why do we care if there's a bend in the chain or not? Well, it actually has a big impact on your health. Saturated fats tend to be much more sticky, solid, greasy. Like I usually think of like butter, bacon grease. They're all solids at room temperatures. But the saturated fats are liquid. I think of like vegetable oil, olive oil. But we'll look at that in a second. But now you understand the difference between a saturated fatty acid and an unsaturated fatty acid. So let's look at what this would look like if it was an actual fat. So right now, these are just called fatty acids, and it's just one chain, right? But what would a fat, like an actual fat, look like? This would be called a triglyceride. If you break apart the word, 3, and then glycerol, triglyceride. So what this looks like is it looks like a, you've got this glycerol backbone, okay? Let's go ahead and specify. We got 3 fatty acids connected to 1 glycerol backbone. And so what that looks like is we got the glycerol backbone. And then you've got, connected to that, you've got 3 fatty acids. So let's actually draw the, the actual chemical structure first. So glycerol looks like this. The glycerol backbone is composed of carbon atoms. And then connected to those, the glycerol also has these OHs coming off of it. And so what we're going to do is we're going to take, so let's label this as glycerol. We're going to go ahead and take 3 of these fatty acids, and we're going to bring them down and connect them in these 3 places. So if we draw that, we have our hydroxyl. So there's 1 fatty acid, and that's going to form a reaction right here and connect. Another fatty acid right here will come in and connect to the middle part, and then the third one will come in down here. And that's how we get right? It's called a triglyceride. So if these join up and they react, they will connect and form this structure that kind of looks like this. Just the overall structure is the glycerol backbone, connected to 3 fatty acids. So if you want, you can label this as a fatty acid. You can label this as a glycerol. 3 fatty acids connected to 1 glycerol, and that's what forms a triglyceride. And most fats that we consume in our diet, most fats are made of triglycerides. Any questions about that so far? How's your guys' brains doing? A little bit fried at this point? Hanging in there? Can we do a little more? It'll be okay? All right. We got a couple more macromolecules to discuss here. All right. There's another lipid that we need to talk about here called a phospholipid, and this is actually what makes up the cell membrane. So you got a phosphate head, phosphate head that is polar, and then we have a lipid tail that's non-polar. That's all we need to say about those. We'll get many more details about phospholipids when we get to Unit 2, which is the very next unit, actually. Anyway, just know that phospholipids are also composed of these chains, right? So you got the zigzag. So this would be like I guess this would be an unsaturated, and then this one that has the bend would be saturated, and then this is an unsaturated. Got that right. And that's why this tail here has a kink in the tail. It's got a bend. It's unsaturated fatty acid. Okay, we're going to move on to nucleic acids now. This one is simpler, I think. All right, nucleic acids. So this is macromolecule number 3. Okay, so it is they are the building block. They are the building blocks of DNA and RNA. A couple things to remember. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So just in case you were wondering what the acronym stands for, that's what it stands for. We're going to keep it really simple today. You're going to learn many more details about like the structure of DNA later, but today I just want you to know that the monomer of these things monomer. The monomer is the nucleotide. The polymer, so once you join the monomers together, you would form a DNA strand or an RNA strand. And you guys know this, DNA is composed of repeating parts. It's just a bunch of nucleotides connected together, a big long chain. So what we're going to do right now is we're just going to look at the monomer. We're just focusing on the monomer, which is the repeating part. So the nucleotide is composed of 3 parts. And that is a phosphate, a sugar, and a nitrogenous base. And it looks like this. I want you to think of like a little house, okay? There's a house. Off of the eave of the house is a sun. And then off of the other eave of the house is a garage. The house is the sugar. The garage is the nitrogenous base. And by the way, that would be like the letters as we know them, right? G, C, A, T, and maybe uracil if you've got RNA. Is this bringing back memories from freshman year, freshman biology? So the nitrogenous base, that's like, guanine or whatever. And then this is, of course, the phosphate. Now, later on, I will show you how these connect to form a DNA strand. But for today, I just want you to know that the monomer is the nucleotide and it's composed of these 3 parts. Keeping it as simple as we can for now. Any questions about nucleic acids? Nucleotides? Yeah, alrighty. Good stuff. We got one more. That is proteins. Proteins is the most complicated because it's the most variable. If you think there's a lot of carbohydrates, just wait till you see proteins. Like with proteins, you can have almost any shape, almost any structure, almost any function. In fact, proteins, they do a huge variety of things. And the reason for this is that proteins can form really complicated 3-dimensional structures, very, very complicated structures. So we'll go ahead and jump in here to proteins. So make some space for proteins now. And let's just check the time, 12:47. Okay, we're doing good. We're gonna make it. All right. Wow. Okay, hang on a second, guys. Sorry, I might have misplaced my notes. Son of a gun. Where did it go? Okay, it's fine. I remember what I wanted to tell you. All right, proteins are, this is really weird though. I'm missing like the most important stuff. Proteins. All right. Okay, couple fun facts about proteins. They are they are made of amino acids. So the amino acid is the monomer, right? And then the protein is the polymer. There are 20 types of amino acids. Each type has its own shape and properties. For example, some are polar. Some amino acids are polar. Some are non-polar. Some are charged, while others are neutral. So there's a lot of variability in these 20 amino acids, and that's actually what gives us the variability in the types of proteins we can make. So let's look at the kind of the generic blueprint for an amino acid. So every amino acid has something called an amine group over here. And notice, by the way, I actually really want you to pay attention. This is the very first time today. This is the very first nitrogen atom that I've shown you. You see that? It's the first time you've drawn the letter N in a molecule. That's significant. And the reason that that's significant is because it's an atom that sometimes is hard to get. And so nitrogen is actually a nutrient. You can think of it as like a nutrient. Sometimes the growth, like, have you guys ever fertilized your garden or fertilized the lawn out front? You know, one of the main ingredient in fertilizer that you spread on grass is nitrogen, because it's very often grass is deficient in nitrogen. So you add the nitrogen and it gives it the ability to grow. Right. So there's nitrogen, and the nitrogen connects to this middle carbon atom, and then the carbon atom connects to something called an R group. Okay, now R is not a real thing. This is this is the variable portion of the amino acid. So what I mean by that is that when you look at the amino acids, see if I can bring why is it not amino acid? All right, it's not letting me type for some reason. Anyway, we'll look it up in a minute. But if you look at the chart of the amino acids, this R group, this is the part that changes between the amino acids. Like, for example, it could be a chain, it could be a ring, it could be some kind of polar thing, some kind of non-polar thing. So just that gets substituted in for here. Right. That's the variable portion. And then over here, we've got a carboxyl group, which is the carbon with the double bond to the O and then the hydrogen. We've seen that before. We've seen the carboxyl group before. Just to remind you, we saw the carboxyl in the fatty acid. Fatty acid was a chain connected to a carboxyl group. Up here, we just have a hydrogen. One other thing to point out, notice that carbon always, remember what we said, carbon has 4 valence electrons, which means it always forms 4 bonds. Let's verify that. Carbon, 1, 2, 3, 4. Check. Carbon, 1, 2, 3, 4. Check. Carbon always forms 4 bonds. And that's again, once again, that's one of the features of carbon that makes it, it lends itself to a lot of interesting shapes and structures. Okay, so this whole molecule here is called an amino acid, right? And it is the monomer. It's the repeating part. that forms the protein. Proteins are highly variable in structure and function. The list is like a mile long of, I mean, you could seriously spend the rest of your life studying different proteins and how their different structures and their different functions and all the roles that they play in the body. Proteins form the machinery and inner workings of the cell. They kind of run everything. They run the show, I guess you could say. So we're talking like all the enzymes in your cells that like help build DNA, do transcription translation, communicate from one cell to another, repair the cell, grow the cell, the checkpoint factors, the growth factors that determine how much the cell is going to grow, when it's going to divide, how often it will divide, when the cell is going to die, and all of those things. It's all controlled by these little proteins. And there's just thousands, hundreds of thousands of different specialized proteins, each of which does its own thing. There's even some proteins that are structural. So for example, your guys' hair, your nails, your fingernails, that's protein. And that's just structural, right? It's not as though your nails, communicate or do anything fancy like that. They're just structural, just like but it's protein. Hair, also protein. Collagen fibers in your body, like your skin, has a bunch of connective tissue called collagen. Collagen's a protein. So there's some structural purposes to protein as well. But not to mention, enzymes, like all the enzymes that do all this stuff, proteins. An enzyme is a protein. Okay. I'm not going to do the notes on. Okay. Can everybody take your reading? Go to page 33, please. And take your pencil or something and a highlighter, pencil. But could you guys do me a favor, and does everybody see where it says primary, secondary, tertiary, quaternary? I want you to star that. Actually, basically all of page 33. So put a star on page 33. And this is me telling you that this is important. Okay. Specifically this page. And I could do notes on the primary, secondary, tertiary, quaternary, but I don't think it's necessary. Not because it's not important. It's extremely important. In fact, you guys will most likely, I'm just telling you right now, you will most likely get a free response question on the test about the 4 levels of protein structure. Take it as a clue here. And the reason I do that is because the chances are pretty high that on the AP test there's going to be questions about the 4 levels of protein structure. All right, any questions, you guys? I'd love it if you guys asked more questions. It makes things more fun. Anybody got anything? Questions? All right. Fair enough. All right, so here's I do have some practice for you guys to do right now. There's a Google form. Go ahead and go to the Google Classroom.